This procedure begins with the homogenization of plant material using ceramic beads and subsequent dichloromethane extraction to obtain the organic phase. Following centrifugation, samples are dried under air and prepared for vapor phase extraction. After elution from vapor phase extraction filters, one can proceed to plant hormone analysis using mass spectrometry and gas chromatography.
Hi, I'm Marie Engelberth from the Jurgen Engelberth lab in the Department of Biology at the University of Texas here in San Antonio.
And I'm Jurgen Engelberth. Today we will show you a simple procedure for the simultaneous quantifications of important plant hormones. We use this method routinely to monitor changes in these hormones in plants under insect herbivore attack. We can also monitor changes in the fatty acid composition.
So let's get started.
For the extraction, you first need to prepare 2 ml screw cap vials. To do this, add 400 microliters extraction solution and 10 microliters of the respective internal standard at 10 nanograms per microliter to a vial. Next, freeze the vials in liquid nitrogen. After freezing, add the ceramic beads to the vials. While the vials are still positioned in liquid nitrogen, add the plant material, between 50 and 200 milligrams. Keep in mind that the weight of the added plant material has to be